Cell cultures. Cell culture is the process by which cells are grown under controlled conditions, generally outside their natural environment. This technique is known as two-dimensional primary cell cultures, and they were first developed by William Roux in 1885. Over the years, several improvements have been done for improve and maintain cells in culture. Primary cells are cells derived directly from living tissues, processed and cultured under optimized conditions. Therefore, they provide an excellent model system for studying the normal physiology, molecular biology of cells, and the effect of compounds. Primary cells have a limited lifespan and will stop dividing after a certain number of cell divisions, so they can be more difficult to culture and maintain than a continuous cell line. Primary cells can be immortalized in different ways, creating a continuous culture that is able to remain alive over time. Cells need to be cultured under controlled environmental and nutritional conditions. These widely vary depending on the cell type. The culture media typically is composed by a basal medium supplemented with appropriate growth factors. It is essential to include an antibiotic in the growth medium to inhibit contamination by bacteria or fungi. Cells are grown and maintained at an appropriate temperature and gas mixture in a cell incubator. Typically 37 degrees, 5% CO2, 95% air for mammalian cells. In vitro or in vivo? In vitro experiments are important as they allow more rapid development of new products, formulation, and only those that appear to be effective go on animal studies. An in vitro experiment is appropriate to study the product on a simplified and controlled environment. In contrast, in vivo studies are needed to see how the animal as a whole will respond to a particular product of substance, for example a vaccine. In some cases, in vitro studies of a product can be promising, but subsequent in vitro experiment may fail to show an efficacy. In vivo studies are done to understand the actual effect on an organism. However, they are expensive and time-consuming. To sum up, in vitro studies are extremely important because they lay the groundwork for further research and represent the foundation of every successful study. My name is Federico. I am a PhD student at Vetagro SPA. I am a biotech graduate, and in my free time I love photography, motorcycling and trekking. Thanks for watching!